Hello everyone. Welcome back to our Plante. In today's lecture, we'll learn all about the life cycle of angiosperms, part 3, in a simple and easy to understand way. So let's begin. The life cycle of angiosperms, part 3, stigma of another flower. The pollination is brought about through various agencies like wind, anemophilus, by insects, entomophilus, or by water, hydrophilus, pollen tube. The stigmas in different flowers are variously modified to receive the microspores. The surface of the stigmas is usually papillate and glandular. The microspores are lodged among these papillae, where they germinate and produce pollen tube. The microspores absorb water and other substances, such as sugars, organic acids, etc., secreted by the stigma and style. The microspores swell up, and exene ruptures at germ pores. The intine protrudes out in the form of pollen tube. The contents of the pollen grains move into the pollen tube. The pollen tube grows through the style and enter the ovule either through micropyle, chalaza, or sideways. The vegetative nucleus lies at the tip of the pollen tube and is thought to be concerned with the growth of the pollen tube. But in some cases, it is present behind the male gametes, while in certain cases, it degenerates. The pollen tube discharge its contents into the embryo sac. The pollen tube and its contents constitute the male gametophyte. Normally, the microspores produce a single pollen tube, monosiphonous, but in some cases, for example, in members of Cucurbitaceae, Malvaceae, more than one pollen tubes, polysiphonous, are produced. However, only one of them develops, and the others degenerate. Sometimes a single pollen tube may branch. The contents of the pollen tube pass to tip of one through the branches, while the others abort, e.g., in Cucurbita pepo. The entry of pollen tube is found in majority of the cases. In majority of the cases, the entry of pollen tube through the chalazal tissue. Such entry is termed as basicamy or chalazogamy. In certain cases, the pollen tube makes its entry by penetrating the integumental tissue. This entry is called mesogamy. Strong gynoecium or pistil is the female reproductive whorl of flower consisting of one or more carpels or pistils. The carpel is an elongated appendage and is a modified folded leaf. The megasporophylls. The carpel consists of 3 parts: stigma, the terminal end of the style which receives pollen grains; style, slender projection of the ovary. The surface of the style may be hairy or smooth. The swollen basal portion is termed as ovary, which may have one or many chambers. The ovary contains one or more rounded or oval bodies. The ovules, integumented megasporangia. The ovule contains a large oval cell called embryo sac, enlarged megaspore. The number of carpels in each flower varies from 1 to many in different plants, but this number is constant for families and serve as an important taxonomic character. Megasporangium, or ovule. The angiosperm ovule has long been considered as a bud, because sometimes a shoot develops from it, and because it was born on placenta, which was considered axial, because from it, as a seed, the axis of the new plant develops. It has more commonly been considered of foliar nature, a sporophyll, a leaflet, a lobe of a leaf, the tip of a leaf, etc. Some are of the view that it is a new structure, an organ sui generis, a complete and independent organ inserted on the stem. However, fundamentally, it is recognized as a structure that bears sporogenous tissue in which megaspore develops. The ovule is termed as an integumented megasporangium. A nucellus is the sporangium, and the integuments are protective structures. The ovules are born in the ovary on a specialized tissue called the placenta, which is supplied with a poorly developed vascular tissue. Each ovule is attached to the placenta by a short stalk known as the funicle. The funicle may be long and curved, or short and stout. The long funicle may be adnate to the ovule body, forming a ridge, the raphe. The place of attachment of the funicle with the main body of the ovule is termed as hilum. The main body of an ovule consists of a mass of sporogenous tissue called nucellus, which is surrounded, except at the tip, by two coverings, or integuments, which arise as outgrowths from the chalazal end. The small opening left at the apex of the integuments is known as micropyle. The junction of integuments and the nucellus is called chalaza. The number of integuments is generally 2. The inner integument is formed first, then the outer one. In some plants belonging to Gamopetalous families, the number of integuments is 1. In certain cases, no integuments are produced, for example in some parasitic families. The presence of one integument is considered as an advanced character, which has arisen either by fusion of two integuments, or by the reduction of one integument, generally the inner one. In a mature ovule, towards the micropolar end, a large cell called embryo sac is present embedded in the nucellus. The embryo sac is actually an enlarged megaspore, which contains 3 small cells towards the chalazal end. These cells are called antipodal cells, while a set of 3 cells known as egg apparatus is present at the micropolar end. The 2 upper cells in this egg apparatus are called synergids, or help cells, while the 3rd lower cell is the egg, or oosphere. In the center of the embryo sac, a nucleus termed as secondary nucleus is present. Variation in general form and in position of micropyle distinguish various types of ovules. All forms of the ovules are modifications of a basic type. Various types of ovules in angiosperms are: 1. Orthotropous or straight. The ovule is orthotropous when it is straight and upright on the placental surface. The micropyle, chalaza, and funicle all are in a straight line, as in members of Polygonaceae, Piperaceae. Anatropous or inverted. The anatropous ovule is inverted and is adnate to the funicle, and the micropyle usually faces the placenta. The ridge formed as a result of the fusion of the funicle and the integument is called raphe. The anatropous ovule characteristics of taxa of Rhanales, Helobiales, and Nymphaeaceae. 3. Campylotropous or curved. The ovule is more or less bean-shaped. The micropyle faces the placenta and is attached near the middle of the ovule. These types of ovules are found in the members of Cruciferae, the Posinaceae, Caryophyllaceae, etc. Amphitropous or transverse. In this type of ovule, the funicle is adnate to the ovule proper, for about half of its length, and the micropyle faces laterally. Therefore, the ovule is placed at right angles to its funicle. Forms of ovules: orthotropous, anatropous, amphitropous, campylotropous, circinotropous. The nucellus and the axis remain in the same line in the beginning, but due to rapid growth on one side, the ovule gets inverted. This curvatures continue till ovule turns completely, and once again the micropyle faces upwards, e.g. in Opuntia, plumbago. Development of ovule. The ovule primordium appears as a protuberance on the surface of the placenta. It grows into a conical structure with rounded tip. This is the nucellus, or megasporangium proper. From the base of the nucellus, 1 or 2 layers of tissues develop, which grow upward and form the integuments. The inner integument develops first, then the outer one. The rate of growth of integuments is faster than the nucellus. Thus they enclose the nucellus completely, except at the tip, forming micropyle. Megasporogenesis. Usually at the time of initiation of integumentary primordia, one of the hypodermal cells, called primary archosporial cell, is differentiated. It is large in size with deeply staining nucleus and dense cytoplasm. This cell divides periclinally into an outer primary parietal cell and inner primary sporogenous cell, megaspore mother cell, or embryo sac mother cell. The primary parietal cell sometimes remain undivided and in certain cases divide periclinally as well as anticlinally to form a mass of parietal tissue which serve as nutritive tissue for developing embryo sac. Various stages in the development of megaspores. The primary sporogenous cell usually do not divide and act as megaspore mother cell directly. It divides meiotically, forming a tetrad of megaspores. Both the divisions are transverse. Therefore, the megaspores are arranged in a line, forming a linear tetrad. In some cases, the tetrads may be T-shaped or inverted T-shaped. The 3 upper megaspores degenerate, and the lowest one, which is towards the chalazal end, develops into embryo sac by enlarging in size. Types of megaspore tetrads: A. Linear tetrad. B. T-shaped tetrad. C. Inverted T-shaped tetrad. There are certain exceptions to the sequence of events mentioned above. In certain cases, allium, the megaspore mother cell undergoes a single division and produces 2 megaspores. The lower one develops into the embryo sac, while the upper one degenerates. In Yucca, Iris, Caltha, 3 megaspores are produced due to failure of the daughter cell to divide. In Lilium, Fritillaria, the megaspore mother cell is directly transformed into a megaspore, female gametophyte. The megaspore, N, is the first cell of the gametophytic generation. The megaspore increases in size into an embryo sac, utilizing other spores and the surrounding nucellar tissue. Such ovules in which the nucellus is completely utilized by the developing embryo sac is called tenuine nucellate, while those ovules where the nucellus is not completely utilized and some of the nucellus persists as perisperm in the seed are known as crasinucellate. The embryo sacs are classified into 3 main types depending on the number of megaspores of the tetrad taking part in the development. 1. Monosporic. Only one of the 4 megaspores of a tetrad takes part in the development of the embryo sac. 2. Bisporic. In some cases, after dyad formation, only one of the dyad divides, the other degenerates. The surviving dyad enlarges and divides to form 2 spores, not separated by a wall. The 2 spore protoplasts within the wall of surviving dyad enlarge and form a bisporic embryo sac. 3. Tetrasporic, where 4 spores are formed but remain unseparated by walls. The protoplast of all enter into formation of embryo sac. Let me know if you need help with anything else. If you found this video helpful, please give it a thumbs up. Share it with your friends. And don't forget to subscribe for more easy biology lectures. Also, add 3 questions at the end of lecture. Strong thanks for watching, and see you in the next class.